The life of a cell is never dull. Whether you're talking about a bacterial cell or a plant cell or maybe a cell from your nervous system, or in fact, any other cell, it doesn't matter. It may not be moving, it may on the surface not really look like it's doing much of anything, but that cell is a hive of activity. Simply maintaining cellular structure requires the continual building and replacing of cellular components, the execution of thousands upon thousands of reactions every single second. And all of this activity requires a constant supply of energy. Now, as we're going to discuss in our subsequent series of videos on cellular respiration, it's important to appreciate that the energy from the nutrients that we consume is not transferred directly to wherever work happens to be taking place in the cell. Instead, it is transferred to a super important molecule, one that all cells use to power almost all of their activity. That molecule is adenosine triphosphate, or ATP. When we look in the cell, we can think of ATP as the energy currency. It is used to drive, to power cellular activity, the building of a protein, the conversion of a reactant into a product, or the transport of a vesicle from point A to point B. And the reason for this lies in its structure. So, let's take a look. ATP consists of the nitrogenous base adenine, a 5-carbon ribose sugar, and 3 phosphate groups. Now, this may look fairly familiar to you, as ATP is a nucleotide. This ATP is constructed using the energy found in the bonds of the nutrients that we consume. Specifically, it's the energy from those bonds that is used to join together the 3 phosphate groups, and it's this region of the ATP molecule that is so important for making things happen. Why is this? Let's take a look at these phosphate groups and notice 2 things about them. Firstly, they're all negatively charged. Secondly, notice how they are all confined into the same small area of the molecule. Well, these charges are going to repel each other, and as such, what they create is an unstable region of high potential energy within the molecule. I kind of like to think of this as almost like a stretched chemical spring. So how is a cell going to take advantage of this? Well, those bonds holding the phosphate groups together, well, they can be broken by hydrolysis. So when reacting with a molecule of water during a hydrolysis reaction, the bond that holds that 3rd phosphate in place, what we sometimes refer to as the terminal phosphate, that can be broken. In terms of our spring analogy, it's like it becomes less stretched. Another way of thinking about this is that those electrons from those 3 phosphate groups are now split between 2 molecules, because the upshot of this reaction is that ATP is converted into ADP, adenosine diphosphate, and an inorganic phosphate. What this actually means then is that ADP becomes more stable, less reactive, and that inorganic phosphate can now be used to power cellular activity. More specifically, what actually happens within a cell is that when that ATP is hydrolyzed, that 3rd phosphate is used to energize a recipient molecule, We call this process phosphorylation. And most cellular work relies upon ATP energizing molecules by phosphorylating them. Why? Put very simply, it makes that recipient molecule less stable and more reactive. So let's take a look at an example of chemical work where the phosphorylation of a reactant is what drives the reaction forward. And let's look at the production of glutamine. Glutamine is produced through a reaction between glutamic acid and ammonia. And what has to happen is that it's the hydrolysis of ATP and the transfer of that phosphate to glutamic acid that puts glutamic acid into a highly energized intermediate state that only then is it able to react with ammonia and to give us glutamine. This reaction simply could not happen without that hydrolysis of ATP and that phosphorylation event. And this same process works for, let's say, mechanical work that a cell has to undertake. When you phosphorylate a protein, what it causes is a reconfiguration of that protein's electrons, and a reconfiguration of that protein's electrons results in a change of shape, or in other words, a movement. If you think about transport work, let's imagine the movement of a particular solute from one side of a membrane to another. Well, that relies upon phosphorylation of a transport protein. The important thing to realize here is that ATP is a renewable source. Cells are continually regenerating ATP through something we call energy coupling. This just means that the energy that is released through the breakdown of organic molecules, something we will cover in our series of videos on cellular respiration, is used to regenerate ATP by joining together adenosine diphosphate, ADP, with inorganic phosphate. And that ATP is, of course, then hydrolyzed and used to power cellular activity. To try and give you some idea of the scale here, the average human cell uses and regenerates about 10 million ATP molecules per second. Over the course of a day, that amounts to about 75 kilos of regenerated ATP. That's a pretty continuous supply of energy for all your cellular needs.